Okay, hi everyone, and welcome again to our class. This time, starting with our second quarter. This is Biology 2, entitled Fundamentals of Biology 2. And as we begin the second quarter, we'll of course be starting with Module 1.1 and then 1.2, and this is entitled Mendelian Inheritance. Now, the objectives for our discussion or the module number one that we have, which is Mendelian Inheritance, would be number one to define common useful genetic vocabularies. Number two, to describe the experimental approach Mendel used to identify the laws of inheritance. And number three, to relate the process of meiosis to Mendel's laws of inheritance. So those are the objectives that we have for the whole of module number one. Now, as we begin with our discussion with Mendelian inheritance, here's a question for all of us. Why do we look like our parents? And of course, probably you have experienced at times where you have relatives or friends, parents, friends, or someone who you've met for the first time and you were there with your parents, and they will say either that you look like more of your father or you look like more of your mother, but that still doesn't explain the question, why do we look like our parents? And Mendelian inheritance will be explaining a lot of this, why we look like our parents, but prior or before before the discovery, or before Mendel made his discoveries about the laws of inheritance, there are several theories or several thoughts on how our traits are passed on from parents to offspring. So there are four of them. Number one is called preformation theory. Number two is theory of pangenes. Number three is blending hypothesis. And number four will be particulate hypothesis. We'll just go through very, very fast here since this is simply an introduction. But take note that during the early 19th century, people do not really have any scientific basis for the transfer of traits from parents to offspring. So starting with the preformation theory, this is quite interesting. People were thinking, since it was a time wherein the gametes have been discovered and the process of gametogenesis are still being uncovered at that particular time, but they know already that the egg cell and the sperm cell are needed to fertilize together in order to produce the offspring. Now, for the preformation theory, it's very interesting that people were thinking or theorizing that a whole person is already inside the sperm cell or the egg cell. So, in other words, for example, the sperm cell would be containing that whole person inside that very, very small sperm cell. Then the egg cell will simply be a nourishing bed wherein once fertilized, then the person inside of the sperm cell will just grow up later on to become the baby, and they will be born and later become the person as it grows to childhood and also adulthood. So it's very interesting. It's called the preformation theory. Another one would be pangenesis theory, wherein the egg cell and the sperm cell will get contribution from the different parts of the body. So for example, here the head will contribute, the neck, and all the other parts to produce the egg cell and the sperm cell. And once they combine together, all of those contributions will be the ones that will be helping to produce the whole person later on as it grows. So that's called the pangenesis theory. On the other hand, there's the blending hypothesis, wherein this is mostly believed in by many people by at that time, is that simply the characteristics of the mother and the characteristics of father will just blend together, just like liquids being blended together as it produces the offspring. You cannot really blame people to think about the blending hypothesis, and that is because as you observe people, the eyebrow looks like the dad, the eyes looks like the mom, the cheekbone or the shape of the cheeks would be looking like the dad, and then the lips would look like the mom. So simply looking at people, you would really, we cannot blame people to think that this is a blending process to produce the offspring. And the fourth one, which is actually an expression of Mendel's experiments and insights would be what calls a particular hypothesis wherein the traits from the two parents are joined together but they are discrete. So they are found on different units or different physical units, and this will just be coming together but still they are separate from each other, and this can be separated again to their original forms. We'll go be going back to that a little bit later, especially as we go towards the study of Mendel. But for now, these are the different kinds of things or ways by which people, especially before the 19th century, are thinking of how characteristics and traits are being passed on from parents towards the offspring. Okay, now with that introduction, we have to now go to the vocabularies that are very important for genetics. As we already mentioned a while ago, these are foundational information for us to understand the whole concept about genetics and especially to answer the questions given in genetics. First of which would be the word genetics. It's described as the branch of biology that deals with heredity and variation of organisms. Two important words that you have to take note: the word heredity, and heredity will give us the idea of how of passing the traits from parents towards the offspring. So that is what we call as heredity. Variation, on the other hand, would be the differences of genetic. How is it? These are the differences in how people look like or organisms look like, and this will be showing us differences among organisms in a particular population or species. Now, we have already discussed this in the first quarter because you are already familiar with sexual reproduction, and the very reason, the very importance of sexual reproduction is to produce variation in organisms. And the reason that variation in organisms would be very advantageous is because, as we have already learned from the first quarter, that if suddenly the environment changes, there can be a bit more assurance that at least a few of the organisms will probably or highly likely survive because of the differences and the variations of organisms compared to each other. So, for example, if through a sexual reproduction you produce clones of the same individuals, or they will be clone of each other, they have the same characteristics. If the environment suddenly changes, then what will happen is that they will all be dying because of the sudden change of that environment. As compared to differences and variation of characteristics, there is a possibly higher possibility that a few of those will remain even if the environment suddenly changes. So genetics, heredity, and variation would be two important words you have to remember. Another word that you have to take note, as we have already studied before, is the word chromosome. And the chromosomes can be defined as the carriers of the heredity information, which we call as the genes. So you're very familiar that the DNA produces the RNA, and the RNA will be producing the proteins, and the proteins would be the ones that would be producing the physical characteristics, traits of a particular organism. We're dealing here what we call as the central dogma of molecular genetics, molecular biology, or genetics, whichever you would prefer. So DNA to produce RNA proteins, and the DNA would be the molecule that makes up the chromosome. So chromosomes are carriers of heredity information, also called as genes. Okay, you have to take note as well that the chromosomes occur in pairs, as you already know, and then new combination of genes occur in sexual reproduction. In the first quarter, we already studied that variation, genetic variation, can happen in two places. Number one is during prophase 1, wherein the homologous chromosome will have form a synaptonymal complex, and this will be producing what we call as recombination. So this will causes variation or reassembling or reorganization or recombination of the genes present between homologous chromosomes. And then later on, when during the metaphase 1, wherein the homologous chromosomes will be pairing together and they will be randomly distributed towards opposite poles to form the gametes. So you can see here that this chromosome, homologous chromosome, can be here, but they can also be randomly distributed so that this one will go to here and here, and they will go to opposite poles. So that will be another layer of what we call as genetic variation. So these are the chromosomes that we have to take note of. Other terms that we have to take note of is gene is the unit of heredity, a section of DNA sequence encoding a single protein. So a stretch of DNA, and there will be sequences in the DNA that will be coding for RNA and later a protein, and that is what you call as a gene, a unit of heredity. Another one is genome, and when we talk about genome, that's the entire set of genes, the entire set of DNA that can be found in all of the cells of a particular organism. So we have your gene and we have your genome. Another one would be alleles. Now, in this case, alleles would be different forms, different flavors, or different alternative forms of a particular trait. So, for example, if you're very familiar with the hairline, you can find people who have what we call as widow's peak, while those others without widow's peak, they will only have a straight hairline, just like me. I have a straight hairline here. There are only two forms of this, which would be called the widow's peak and the non-widow's peak. And we're talking about that type of hairline, that's the particular character. So when we say alleles, these are flavors or different forms or different alternatives of a particular characteristic. Another example would be this: the earlobes. There will be people who have earlobes that are unattached, or what we call as free earlobe, and there will be earlobes that will be attached. So you will be noticing here these are the character or the trait would be earlobes. What kind of earlobes? And then there are two forms of this. One is free earlobe unattached, while the other one would be attached as if it's attached towards the neck. So these are what we call as allele, two genes that occupy the same position on homologous chromosome that will be covering the same trait, but they express different forms or different flavors as mentioned a while ago. Okay, you have gene, genome, allele, and another one is called locus. Particularly, the locus would be defined as the fixed location on a strand of DNA where a gene or one of its allele is located. Now, looking at this picture, we have two homologous chromosomes here, and then you will be noticing that there are different bands, and each of the band will be symbolizing a particular gene. Again, the gene is a unit of heredity, and later it will be producing a protein. The DNA produces the RNA and then produces the protein. So these are the different genes. You have the D gene, the E gene, the F gene, and the G gene, and also the H gene. Now you will be noticing here that there are two forms, the big D and the small d, which we will be discussing a bit later. But so this will be simply showing or symbolizing to us that a particular gene can have several flavors or several forms, but for the Mendelian inheritance, we are looking at two forms of this, so two different alleles or two different flavors, and then the loci would be the particular placement or where the gene can exactly be found. So for the F gene, it will be its loci is very near the centromere. For the G gene, it will be found in the middle of the arm, while the H gene, the loci will be found on the end of the arm. So those are important things that we have to take note of. Now here's a question that I would like us to ask. What's the difference between dominant allele and recessive allele? We were talking about a while ago the different forms of the particular character, and we also showed a while ago that there's a capital B and a small b that symbolizes the two alternative forms. So this would be a symbol for dominant allele and recessive allele. So what would be the difference between the two? Well, as you can see here, the dominant is the character, is the allele that will always be expressed when it is present, while the recessive is only expressed when no dominant genes are present. In other words, this is the allele that masks the expression of the other allele, while the recessive would be allele that is the one that is being masked by the dominant allele. And so the big A or the capital A will be symbolizing the dominant allele, while the small a will be symbolizing the recessive allele. Okay, so again, the dominant would be the one suppressing the expression of the small a or the recessive, while the recessive can only be expressed when only the recessive genes or alleles are present. Now, with this, just take note that in genetics problems, we always use a capital letter to symbolize the dominant gene, whatever trait that may be, and the small letter will always be used to express or to symbolize the recessive gene. Now, since organisms are diploids, we would be expecting that there will be one allele and another allele that will be found together in a particular diploid organism. And that leads us to the fact that there can be three possible combinations of these alleles. Now, this would be what we call as dominant and dominant, dominant and a recessive, and the other one is both of them will be recessive. So these are the three possible combinations of what we call as the different alleles. So a big R and a big R, a big R and a small R, and a small R and a small R. So those are the three possible combinations. Now, before I forget, there's a picture that we were not able to take note of, and this would be what we call as the alleles a while ago, as mentioned here. So this would be the allele, for example, for purple flowers, and this will be the allele for white flowers, but they are found in the same loci, in the same position of the chromosome, and they will be talking about one particular trait, and that will be the specific color of a flower. Now, interestingly enough, you will take note that oftentimes, of course, these alleles will be very different from each other. So, for example, here, the only difference is a C, and the other one will be an A, and this will can produce what we call as alternative forms of a particular trait. Because they have the same gene, but they differ on certain bases, that will be producing one the purple color and the other will be the white color. So they are the same. They are different forms of an allele, different forms of allele of the same character that we're talking about. Okay, going back here. So we have three possible combinations: the bigger the big R, the bigger the small r, the smaller the small r. Two dominant alleles together, one dominant, one recessive, and two recessive alleles together. So you will always be finding these kinds of combination, especially in Mendelian inheritance. And later we'll be talking about monohybrid crosses, but we'll leave that for a while. So these are the symbols that you always be meeting in genetics questions. So you need to be very familiar with these symbols, which I am very sure you are already very familiar with. Now here's another question: which one would be the expression of this? So as you can see here, since the big R is dominant, if you combine it with a big R, you will always have a dominant phenotype. If you have a big R and this would be combined with a small r, you will have here as well as a dominant phenotype, while the small r and the small r, when they are combined together, they are what we call as a recessive phenotype. Now, we have a hand expression so that we can easily remember that. Imagine that the open hand is dominant, and then you have the closed hand as the recessive. So in a diploid organism, when you combine together the different alleles, if one is dominant, the other is dominant, and you combine them together in the diploid organism, you will see, or the manifestation would be dominant character. If you have a recessive and you combine that with the recessive, you will always have a recessive manifestation of the characteristic. But if you have a dominant combined with a recessive, so what would be the characteristic or trait that is being expressed? It would be dominant, because the dominant will be covering the expression or suppressing the expression of the recessive, but the recessive is always there. It did not disappear, but it is always there. It simply is being hidden by the dominant. So that would be the big R and the small r. Okay, so those are important characteristics. Terms that you have to take note of. Additional terms would be this question: what would be the difference between homozygous and heterozygous? You will always be uh, taking note of this, because these words you will always be finding in genetics questions. So what's the answer to this? Well, you already know perhaps the answer to this is that the homozygous have identical alleles, while the heterozygous would be having different alleles for a particular trait. Going back to our picture a while ago, so since the big R and the big R are of the same type, and they are the dominant genes, you will call it as homozygous, particularly homozygous dominant. If you look at the small r and the small r, they are the same allele together. So what this means that they are homozygous, but particularly homozygous recessive will be the term used for them. But for the big R and the small r, they are different alleles, different forms of allele together. So you call that as a heterozygous. So homo means the same, hetero means different, and that will give you a basic idea. Okay, so that's another set of vocabularies that we need to take note of to help us in genetics problems. So if this will be expressed in chromosomes, you have here the chromosome, another chromosome, and since both of them have the big A, that's why it's called big A, it will be called a homozygous dominant. On the other hand, if this would be one of the homologous chromosome contains the big B, while the other homologous chromosome contains the small b, since they are different, then they will be called as heterozygous. So very easy for us to take note of. Another terminology, what's the difference between phenotype and genotype? We were mentioning, we have already actually mentioned, we have actually described this a little bit a while ago. So the difference between the words phenotype and genotype would be the genotype is the genetic makeup of the organism, while the phenotype will be the physical appearance of an organism. Genotype plus the environment. In other words, if you will be looking at the keyword geno, this will be pertaining to the genes. It will be talking about letter combinations, as you, if you will, the letter combinations. So it's a big A, small a, or small a. So we're looking at the genotypes and the letter combinations of that. Is it homozygous dominant? Is it heterozygous? It is homozygous recessive. But when we're talking about phenotype, we are talking about here the physical characteristic of that particular organism. So for example, we're talking about here the widow's peak and the non-widow's peak. So that's phenotype. We're talking about here the dimpled and the non-dimpled, then we're talking about here the phenotype. But if we're going to be talking about what's the gene, what's the allelic combination or the letter combination that produces the widow's peak, now we're talking about genotype of this. So here's a picture of that. So the phenotype would be the set of observable physical traits. So you have your purple, and then your purple, and the white. So this would be the physical manifestation or phenotype coming from the pheno, which is related with the word physical, and genotype, which would be related with the word genes. You now have to take note of the letter combination, the genetic constitution, or is it homozygous dominant, homozygous recessive, or is it heterozygous? Okay, so that would be two more important vocabularies. Last of the vocabularies that you will be needing, at least as a foundation for genetics question, would be this, the P, the F1, and the F2. Now, of course, in genetics, you have to take note that there will be a lot of process. We'll be dealing with monohybrid crosses and dihybrid crosses a little bit later. But take note that you have to take note that there are many crosses that will happen. So take note, you have to remember that the first organisms or parent or pairs of organisms that you cross together will be called the parental generation, while the children of the parental generation will be called the F1, which is the first filial generation, offspring of the genetic cross, the first genetic cross that you made, the original genetic cross that you made. And then if you have the F1 generation and you get another F1 generation that cross them together, the children that will be produced on the next generation is called the F2 generation, which would be the second filial generation of a genetic cross. Now, this is very important for you to remember because a lot of the questions will be pertaining to P generation, F1 generation was crossed with another F1 generation. What would be the ratios of the F2 generation? That is why you have to take note of all of this stuff. Okay, and this will be the end of the video for vocabularies. We will continue with Mendel's law on the next video.